Welcome to this explainer. Today, we're taking those textbook diagrams of the human transport system and throwing them right into the real world. We're breaking down these complex biological mechanisms so you don't just memorize them for a test, you truly, fundamentally understand them.
Here's our roadmap for today. We'll start by tackling an actual exam question on blood cells and balloon angioplasty. Then we'll zoom out to circulatory system types, heart structure, blood clotting and groups, and finally, wrap things up with the lymphatic system. Let's get right into it.
Section 1. Exam Question 8, Blood Cells.
So imagine you're staring at this exact scenario from the Sellingar trial paper. You've got to identify blood cells just by looking at their structures. It asks for the function of cell T. Notice how it isn't even a full, round cell, it's just a tiny fragment. That's a massive clue. It's a platelet. It's literally just a piece of broken-off cell cytoplasm. And its whole job? Strictly blood clotting. It's the frontline defense to stop you from bleeding out when a vessel gets damaged.
Now, if you want full marks in the marking scheme when comparing cell R and cell S, you need to state a clear similarity and their differences. The easy part? The similarity. They're both white blood cells, also known as leukocytes, which are your body's pathogen fighters. But check out the differences. Cell R has this grainy, granular cytoplasm and a weird lobed nucleus, making it a granulocyte, specifically a neutrophil. Cell S, totally smooth, non-granular cytoplasm, no lobed nucleus. That makes it an agranulocyte, like a monocyte or a lymphocyte. Honestly, nailing these visual cues is the absolute secret to acing these exams.
All right, moving on to Section 2. Exam Question 8, Balloon Angioplasty.
You hear about coronary angioplasty a lot when people talk about cardiovascular disease, right? Well, here's how it actually works. Your coronary artery is the VIP blood vessel responsible for supplying oxygenated blood directly to your heart muscles. But when it gets choked up with plaque, flow is severely restricted. So doctors literally insert a tiny, deflated balloon into the artery and inflate it to temporarily widen that blocked space, the lumen. Then, a little wire mesh tube called a stent expands with the balloon and stays behind like a scaffold to keep the artery permanently open. It's an absolute lifesaver.
So why does this matter so much for the patient? Well, by physically wedging that artery open, balloon angioplasty immediately restores vital blood flow straight to the cardiac muscle tissues. This means a huge, instantaneous boost in the transportation of oxygen and nutrients. And the ultimate goal? It directly prevents the risk of myocardial infarction. That's just the clinical term for a heart attack, which happens when heart tissue literally starts dying because it's suffocating for oxygen.
Section 3. Circulatory system types.
Let's zoom out to the broader textbook theory for a second. There is a totally stark contrast between open and closed circulatory systems. Take an insect, for example. They have an open system where a fluid called hemolymph pumps directly from the heart into the main body cavity, the hemocole, just completely bathing the internal cells. But for humans, fish, and amphibians, we've got a closed circulatory system. Our fluid is blood, and there's no splashing around freely here. It is always securely contained within a continuous, closed loop of blood vessels.
Focusing in on human blood. If you took a vial of your blood and spun it down in a centrifuge, you'd see it's about 55% blood plasma. The other 45%? Mostly erythrocytes, your red blood cells. Now, these guys actually ditch their nucleus as they mature so they can cram in as much hemoglobin as possible. That's how they carry oxygen around as oxyhemoglobin. And taking up less than 1 tiny percent of that whole vial are your pathogen-fighting leukocytes and those clot-forming platelets we just talked about.
Section 4. Heart structure and beating.
Okay, before we trace how the electricity flows, we have to talk about this absolutely wild property of the heart. It is myogenic. This means your cardiac muscles contract and relax totally on their own. They don't wait around for a signal from the brain or nervous system. Literally, if you put living heart tissue in a warm, oxygenated, nutrient-rich solution, it would just keep beating rhythmically all by itself. How cool is that?
So here's the specific electrical pathway that coordinates that contraction. It kicks off at the sinoatrial or SA node right up in the right atrium. Think of this as your heart's built-in natural pacemaker. It fires off electrical impulses that make both atria contract. Boom. Next, those impulses hit the atrioventricular, or AV node. From there, the signal shoots down the center septum through the bundle of His, and finally spreads up around the bottom through the Purkinje fibers at the apex. This causes the ventricles to squeeze powerfully from the bottom up. And when those heart valves snap shut during that coordinated squeeze, that's exactly where you get that classic lub-dub sound through a stethoscope.
Moving right along to Section 5, Blood Clotting and Groups.
Looping back to that first exam question about the platelet, let's look at the actual chemistry of blood clotting. When platelets in damaged cells pile up at a cut, they release an activator called thrombokinase. Now, using calcium ions and vitamin K, that thrombokinase converts an inactive plasma protein called prothrombin into a highly active enzyme called thrombin. Thrombin is the real catalyst here. It converts a soluble protein, fibrinogen, into an insoluble protein called fibrin. This fibrin forms a literal, physical thread-like net right over the wound, catching red blood cells like fish in a net and sealing the break.
Beyond just clotting, another absolutely crucial blood factor is surface antigens. An antigen is basically a chemical ID tag sticking right out of the surface membrane of your red blood cells. Based on the genetic lottery, you might have antigen A, antigen B, both of them, or neither. You also have to factor in antigen D, which you probably know as the rhesus factor. If you have it, you're rhesus positive. Pinpointing these exact ID markers is non-negotiable for safe blood transfusions, and even for managing pregnancies, so you don't trigger a massive life-threatening immune response.
Because if you mix the wrong blood types, you get agglutination, which means the blood literally clumps together inside your veins. Bad news. But look at how the compatibility breaks down here. Group AB blood has both antigens but zero antibodies against them. That makes them the universal recipient. They can safely take blood from anybody. On the flip side, Group O has zero surface antigens at all to trigger an attack, which makes them the universal donor. They can give blood to anyone.
Finally, Section 6. The lymphatic system.
Okay, so when high-pressure blood rushes into your capillaries, fluid physically gets squeezed out into the tiny spaces between your cells. We call this tissue fluid, and it bathes your cells in nutrients. Now, about 85% of this fluid gets sucked back into the blood at the venule end. But what about the leftover 15%? It drains into these one-way tubes called lymphatic capillaries, and once inside, we call it lymph. The big difference? Compared to regular tissue fluid, lymph is packed with dietary fats absorbed straight from your digestive tract, and it has a massive army of pathogen-fighting lymphocytes.
So what happens if this delicate plumbing system backs up? Say the lymphatic vessels get blocked, or maybe you're totally sedentary and your muscles aren't squeezing to push that lymph fluid along. Well, that leftover 15% gets trapped. When this fluid return system fails, all that excess tissue fluid just accumulates and builds up in those spaces between your cells, causing a severe, painful swelling condition known as edema. It's a pretty stark reminder of just how perfectly balanced our internal transport networks need to be.
Which brings us to a final thought for you to chew on. Knowing exactly how these incredible biological transport systems keep us alive, from your myogenic, self-beating heart to the delicate cascade of clotting factors, to the silent drainage of your lymphatic system. What daily habits are you going to change today to make sure your internal networks keep flowing smoothly? Because hey, understanding the biology is one thing, but actually applying it to your own life, that's the real test. Thanks for hanging out with me for this explainer, and keep exploring the amazing science inside you.